Hello, everyone. We're glad you've joined us for today's live webinar, Overcoming the Clinical Sample Bottleneck for Expression Studies, presented by Drs. Mark Manary and Sean Diehl. I'm Christina Joel of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning, and sponsored by Affymetrix. Affymetrix technologies enable multiplex and simultaneous analysis of biological systems at the cell, protein, and gene level, facilitating the rapid translation of benchtop research into clinical and routine use for human health and wellness. Affymetrix, part of Thermo Fisher Scientific, provides leadership and support, partnering with customers in pharmaceutical, diagnostic, and biotechnology companies and leading academic, government, and nonprofit research institutes in their quest to use biology for a better world. To learn more, please visit www.affymetrix.com. Okay, let's get started. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions to the speakers during the presentation while they're fresh in your mind. To do so, simply type them into the Q&A box, which will open when you click the green Q&A button at the lower left of the presentation window. Once Dr. Manary and Dr. Diehl finish their presentations, we will open it up for a live Q&A. To enlarge the slide window, click on the screen icon in the lower right-hand corner of the slide window. If you experience technical problems seeing or hearing the presentation, just click on the support button found at the top right of the presentation window, or you can report your problem by clicking on the green Q&A button at the lower left. We have two speakers for you today, Mark Manary, MD, and Sean Diehl, PhD. Dr. Manary, an American pediatrician who has worked in sub-Saharan Africa for most of his professional life, was educated at the Massachusetts Institute of Technology and Washington University School of Medicine in St. Louis. He runs a basic science lab in St. Louis and maintains 3 active field research teams in Africa, in Sierra Leone, Malawi, and Ghana. Dr. Manary is a Helene Robertson Professor of Pediatrics at Washington University and has published more than 175 peer-reviewed articles. Improved gut health has been his primary focus in the past 5 years. Dr. Manary's professional goal is to fix malnutrition for kids in Africa. To this end, he has developed ready-to-use therapeutic food, a novel lipid-based food that has been accepted as a standard care for uncomplicated severe acute malnutrition by the UN agencies. He continues to explore the basic pathophysiology and metabolism of malnutrition, and has developed a new diagnostic method to measure host transcript in feces using sensitive microarray and PCR methodologies. Dr. Diehl is an assistant professor in the Department of Medicine Infectious Diseases and associate director for research for the Vaccine Testing Center at the University of Vermont. He received his PhD in cell and molecular biology from the University of Vermont and did his postdoctoral work at the Academic Medical Center of the University of Amsterdam in the Netherlands. He has published 30 peer-reviewed articles and serves as a reviewer for NIH and the Congressionally Directed Medical Research Program. At the University of Vermont Vaccine Testing Center, Dr. Diehl's overarching research goal is to develop novel indicators of how vaccination triggers the immune response to promote durable immunity and protection from disease to develop novel correlates of protection, particularly for infections of global importance such as Dengue and Zika viruses, rotavirus, and other pathogens. She's also been investigating how conditions such as malnutrition impact the infant immune system and has recently developed a workflow that allows for HTA 2.0 microarray analysis of field-preserved small-volume pediatric whole blood samples. The speakers' complete bios are found on the LabRoots website. Dr. Manary will now begin his presentation. Dr. Manary?
Great. Thank you so much, Christy, for the kind introduction, and I'm delighted to have the opportunity to talk this morning. What I'd like to talk about is using the human transcriptome as elucidated by a microarray to diagnose a very elusive condition called environmental enteric dysfunction. A huge mouthful of complicated words there, but we're going to go through them and help you understand exactly what this is all about. We're going to talk about stunting, which is child enemy number 1, and environmental enteric dysfunction as a cause of stunting. The whole transcriptome analysis in feces is possible and reliable, and show you that methodology, and interpret it to define the pathobiology, and then how we can use a focused PCR technique, which was driven by the microarray as a biomarker for this condition. So, as we get started, stunting is global enemy scourge, global health scourge number 1. It affects roughly 30% of children worldwide under 5. It refers to, in a literal way, having short stature, but it really implies a whole host of deficits which are overall devastating. So we're talking about counting for 21% of disability left adjusted life years among all kids. We're talking about something that predicts poor cognitive function. For kids in Africa, stunting affects about half of rural children, and Africa is a rural continent, remember. Africa is half, 60-70%, excuse me, of kids are stunted. Sorry, we've had some technical. Okay, I'm back on here. Okay, as far as stunting goes, the stunted children are less likely to attend secondary school. In their life, they're going to earn 22% less in terms of their income. Their life expectancy, 17% less, and this costs them 8 IQ points. So overall, this is a huge, 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 huge problem. And when we think about growth and development, the paradigm that I've got here is a 3-legged stool. And on this platform, supported by 3 legs, you have good growth and development. You need each leg to be strong and sufficient in terms of the to allow for good growth and development. And 2 of these legs are things that we've known a lot about for a long time. The one on red on the right is a diet that provides adequate amounts of micro and macronutrients in bioavailable forms. The one on the left, the blue one, it requires that you don't have an excess of common infections. And these common infections are not some kind of chronic infections or chronic illness, but they are things like malaria, diarrhea, lower respiratory infections. But if you just have too many of these, you don't grow at the same time you're having an infection, so you won't grow and develop. And then the 3rd leg here, which is kind of mysterious, on the green side, is called good gut health. So, what do we know about good gut health? Well, I would say that a year or 2 ago, we would show you these pictures that you see here. And on the right side, you see poor gut health, and on the left side, you see good gut health. You see something that looks different. You see the shape of the villi are in the good gut health are long, and the shape on the right side with the poor gut health are blunted, reduced surface area. The working surface area of the gut is that absorbs nutrients is decreased. If you stain these things for immunological cells, you'd see that there's a lot more inflammatory cells in the bad gut health side. But really, that's fairly vague, and what causes that, or how we can affect that, what pathways are driven by that, how we can detect that, are all fairly mysterious to us until we started using the microarray. Okay, so the way gut health is measured at present is something called a lactulose mannitol test, which is a dual sugar absorption test. In other words, these sugars that are not metabolized, we get children to drink these, and we look for these sugars in the urine because they're not metabolized. So one of the sugars is absorbed across the surface area, the mannitol, the monosaccharide, and that gives you a sense of how much the surface area of the gut is. The lactulose, which is a disaccharide, is leaks in between cells and paracellular junctions, so that the lactulose is a sign of the integrity of the cells and the cell adhesion. And this is the standard test, then, way to measure poor gut health. And it is, you can see here on the left, the bar graphs show you that if you have a good lactulose mannitol test, in other words, low lactulose, high mannitol, then you have good growth. On the y-axis here, we're measuring your linear growth. And if you have some moderate disability, you have some compromised growth, but still good. But if you have severe EED, then you would have reductions in your linear growth, such that you would be losing compared to your counterparts. And the numbers there on the right are meant to show you that in different kind of modeling exercises that the LM test is a very important predictor of linear growth. So what we did, though, is we decided to took a population for investigation, and these are rural African kids, and it was alluded to in the introduction that I maintain 3 field teams and about 150 employees, and spend actually most of my time working in Africa, although I'm employed by Washington University, to look at what EED is. And you can see here on this slide near the bottom, these are kids who are under 5. These are kids who, about 20% we would say have good gut health, and 20% have severe EED, and most of the kids are moderate. And this is typically what we'd see in rural Africa. More than half of the children are affected by this poor gut health. Typically the way they live, these are folks that live in mud huts with grass roofs. They don't live in electrified structures. They often have domesticated animals that they keep in their homes at night for safety. Improved homes might have a use of a pit latrine or a borehole for water source. So unfortunately, a very common a living scenario for kids in Africa, and really important factors associated with poor gut health. So we wondered if we could use stool samples to find out what the body was doing in environmental enteric dysfunction, okay? So first we have to wonder about, can an RNA, a messenger RNA, survive, and is it present in stool samples, and does the stool have to be preserved in any special way right away? And that's what this graph is showing you on this slide. There have the colored dots are all the very same sample that has been flash frozen at different points in time from 0 to 24 hours. And on the y-axis, you can see 2 different RNA transcripts, toll-like receptor 4 divided by GAPDH, which is a housekeeping gene. And then you can see that these dots overlay each other pretty well. So in other words, if you get a stool sample and you preserve it by making it cold in 4 hours or 8 hours, that's probably quite adequate for RNA. And even though the RNA will degrade a bit with time, the ratio of 2 RNAs in the sample remains pretty constant. So that was encouraging to us to say that this particular kind of biological sample was relatively stable and wasn't going to be greatly affected by the way we collected it. And if we look at the preparation that we used, we'll go ahead to that slide to start with here. If you look at the pie chart over here, you look at the RNAs in stool, in human stool, and you see most of it is bacterial, and most of it is ribosomal. In fact, only 1/4% are coding host RNAs. So we are looking for a needle in a haystack, okay? So whatever method we use to isolate this RNA, we're going to have to be very conservative. We can't throw half of it away because there aren't very many transcripts in there to detect. And this method over here shows you how we were able to do that. You're not really need to view the details here, but we're prepared to answer those questions. This has been put forth a couple times in a peer-reviewed publication as well to share this methodology. But it's a little bit unstandard. It's not how you would say prepare a sample just PCR. So we use some differential centrifugation and extraction on a bead-based technology. Okay, if you were going to look at the transcriptome agnostically, you would probably, in this day and age, you would look at RNA-seq or a high-seq procedure. So this basically takes all the transcripts in the stool, of which you can see on the graph, mostly of them are bacterial, and amplifies them and detects them. Okay, so that is something we tried with fecal samples, and we were utterly failed at that. We just cannot distinguish any meaningful differences in there. And bear in mind, I didn't emphasize it, but when I showed you that picture of good and poor gut health, that's the upper intestine. Most things that come in the feces are from the colon and the lower intestine. So we're really, 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 really looking for a needle in a haystack here with these host transcripts associated with poor gut health. So we turned to the HTA, and we actually had used an earlier version, not successfully, but the HTA 2.0. The advantage of using the microarray is that it is going to preferentially bind human and just let the bacterial stuff wash away. And the HTA array is, as you may learn more about it, is basically a chip with 6 million sites, and each site is a 25-mer, and the 25-mer sort of marches systematically through the human genome, offset by a couple of bases. So, bases 1 to 25, and then bases 3 to 27, and so forth. And this goes through coding, non-coding, the whole genome. So you get a picture of the whole genome. Now there are multiple duplicates on the array. There also are additional sites helping to identify certain important variants in splice junctions. So it's a very complete look at the transcriptome. And then the method which we use for the microarray, that I think is illustrated here on this cartoon. You can see then the preparation was just to isolate the RNA very conservatively and apply it to the array, and then you get all these different luminescence values for 6 million sites corresponding to these 25-mers. In our case, we could detect about 18,000 different genes. And these are not whole genes on the array, but of course they're just fragments of genes. And so there's a way to aggregate these into related sets, and then also the related probe sets are then aggregated into genes. And then once you get these signals, you have to do something with them, and those are choices that bioinformaticists can make. And then since this was very new ground, I would have to say when we did this, and maybe even today, most people are skeptical that you can reliably do anything with feces. We chose 3 different normalization methods. They're RMA, iron, and FARMS, and if you're familiar with those kind of things, they're RMAs are the most common, most standard, and iron is also pretty common. And then we identified transcripts that were associated with our LM test, which is our marker measure, quantitative measure of EED. We looked for differential expression. We looked for correlation coefficients, either Pearson's or Spearman's. And we required that any candidate gene actually be showing up as related on all of these different particular criteria. And then we can use those set of genes that are up or down regulated to map them to biological pathways, either canonical pathways or KEGG pathways, and then we can get a picture of what the host is doing in EED. So, to start with, I would say that we wanted to see that if we ran the same sample twice, did we get the same answer? And that's what this picture shows you. It's indeed true. The blue lines at the top are telling you that if you take a piece of tissue which we know that these microarrays are made to deal with, what do you get? And then the red lines at the bottom are showing you when you take feces. Okay, so the X and Y axis represent the same sample being run on two different microarrays. Okay, so we run the same sample twice, we get the very same answer, reproducible and detectable. This picture here shows you that the pattern of signal intensity between tissue and feces is more similar than it is different. Okay, so this is the shape of these sets of bars is what we're looking at here. And you can also see that the one in the middle, the one normalized by the iron method, that one is most similar, so we often focused our results on iron normalization as well. Okay, so both of these two pieces of information encouraged us that we were getting something reproducible and reliable, so we would then have to learn whether we could interpret this into something meaningful. We used to validate the microarray, we had to use a PCR technique which was quite sensitive called digital drop PCR. And this is a case where you set up a normal PCR reaction and then you sonicate it in oil, and then you get all these little bubbles, and then you little aqueous bubbles, usually about 20,000 of them, and you then count each of those bubbles with your fluorescent probe as either it lights up or it doesn't light up. And that is a very good way to look at low copy numbers of transcripts, which is exactly what we're dealing with. Remember, we're dealing with the needle in the haystack. We're dealing with very low copy numbers here. So we validated this on this slide here. We show you that for 40 or 50 or so different transcripts, that you get the same result. The open bars and the dark bars are very similar in the correlation coefficients between the microarray as well and the ddPCRs of the very same samples. Okay, so now we believe that these are actually measuring transcripts and measuring them reproducibly. Well, we then went on to look at about 260 kids with varying levels of EED, and as I said, EED is going to be a condition that's not discrete. You don't have it or you don't have it, but you either have more of it or you have less of it, or you have a lot of it or you have very little of it, and we quantify that with an LM number. And you can see that we identified several transcripts. Here we have 18 by ddPCR. Looking here on this slide with these 260 microarrays, you can see depending on the normalization method, we identified about 600 or so, 650 that are correlated with the lactulose measurement, that are differentially expressed with numbers of about 150 to 300, and then correlate, and then in pathways that were in up or down regulated, up or down expression, of KEGG or canonical pathways. So this is right away, it's pointing to something that is not a condition of, that would say be like a specific infection. Okay, a specific infection is probably going to have fewer pathways and fewer transcripts upward expressed. The purpose of this heat map here is to show you on the right side there's more red, and that's that there are some transcripts that are expressed more often in children with severe ED, and the right side of it there's some more blue, which some transcripts are not expressed much. And then the picture down here with the list of 12 or 13 transcripts that are most strongly and consistently associated with EED. If you look down what those transcripts do, you'll be impressed that they do, they really engage the whole spectrum of the human immune system. So we see responses to bacterial infections, viral infections, fungal infections, parasitic infections, and this indicates that this EED is not a single infection, but somehow the barrier function of the gut is going to be disrupted, and that's what's causing this problem. And then this picture here shows you the pathways, the canonical ones on the right and the KEGG pathways on the left, that were expression was changed with EED as opposed to no EED. And you can see the types of pathways have to do with immunological activation, but also cell adhesion, barrier function integrity. So this tells you something else about the pathobiology of it. And so we can elucidate this with several transcripts of interest here. And it's not important that you read any of these or all of these, but to know that some of these are involving mucus layer production, mucus layer integrity. Some of them are immune system activation, like calprotectin. Some of them are just enzymes that particularly are expressed in the small intestine, so something that differentiates the small intestine as opposed to the colon. And some of them are for growth of the small intestinal cells. So when I showed you that picture at the beginning about it being fairly opaque, we had no idea what was going on in EED. That was a couple years ago, and after doing this study with the microarrays, you can see the same picture at the bottom here, but now we're able to draw a cartoon above it, and we're able to get an understanding of what's actually going on in EED, which is, as I say, a central cause of stunting worldwide. So you see in the cartoon, on the left, we have normal upper small bowel barrier. We see there's a thick mucus barrier, and there's a layer of commensal bacteria, and those two prevent the contents of the lumen and the microbes in the lumen from interacting with the surface, allowing the surface just to absorb nutrients. The immune cells are waiting in reserve there. But then you move over to the right side of this cartoon and you look at that and you see what's going on in EED. We've disrupted that mucus barrier. We have lots of exposure of enteropathogens to the gut epithelium. We have concomitant breaks in integrity and increased permeability. And the host response that you see written over here on the far right of the slide is what we learned from this microarray technology. That there's recruitment of diverse immunological active cells, particularly viruses. So we often focus in the magic of the microbiota relating to bacteria, but the viruses really seem to be prominent here in EED. And of course, agnostically looking at the host response gives us clues about that, which, say, doing metagenomics or something with bacteria won't. Compromised mucus layer, and then apoptosis and death. So this particular story that I've told you is very, very powerful. So we started this a couple years ago, not knowing really what was causing this, but knowing that it was incredibly important for children worldwide. And now we have some very clear insight into the pathobiology because of what the microarray is able to show us. And we did certainly take the microarray and push its limits. So when we pushed it to a new application that probably wouldn't be on the package insert or something, but we've then since taken that, and with some clinical risk factors, we've identified a biomarker of EED. So that, when we do the biomarker, of course, we are going to do it by PCR. We're not going to run a whole microarray, although you could imagine that a custom microarray would probably be useful in this matter. And you can see we have 6 transcripts down below that, again, represent a diversity of functions in the upper small intestine, the mucus layer. Some Reg1A is a structural protein in this, only in small bowel. Calprotectin is an inflammatory response, as well as TNF. So And that, I think, should bring this to a close. Lots of people to thank in this, and these are always the most dangerous slides, the acknowledgements, because for every name up there, there's going to be 10 that you haven't mentioned. But in particular, we've used this application for defining a new biomarker for the Bill and Melinda Gates Foundation for EED, and we couldn't certainly have done this without the support of Affymetrix, as they gave us many of the microarrays to work with. And we're now currently looking at a large intervention trial with legumes, which the USDA is funding. Legumes because it could be a food that heals this barrier function, and would not be special because these legumes would then be an avenue to preventing stunting, not only by their nutrient content, but what they're doing to the milieu of the gut surface. So I've talked for a long time, so I'm going to stop now and turn it over to Sean.
Thank you, Dr. Manary. I will now hand it over to Dr. Diehl for his presentation. Dr. Diehl?
Everyone hear me? All right. Switching to my slides now. Okay. So thank you all for being here. It's actually a really nice dovetail with Mark's talk. And so again, it's going to relate to HTA 2.0 and new applications for this under the theme of overcoming the clinical bottleneck for gene expression studies. And in particular, what I'm going to be talking about is analysis of gene expression in tissue that's ubiquitously available, not quite to the same level as stool, but nonetheless blood, but presents its own troubles. And that I'll walk you through what we've looked at over the last few months in trying to understand transcription analysis in whole pediatric blood. So just some quick disclosures. Affymetrix has provided a little bit of technical and partial material support. The project has been funded by the Bill and Melinda Gates Foundation and a pilot grant from the Vermont Center for Immunobiology and Infectious Disease. So the background here is, again, understanding some more things about infant survival and thriving. And so across the world, unfortunately, still about 450 children under age 5 die from infections worldwide. Now, vaccines have come a long way in preventing disease, but these regimens are very long, complicated, and are one size fits all, as illustrated by this scheme here from the CDC. And for American children, this is relatively easy to fulfill with access to healthcare and clinics, et cetera. But in rural areas in the developing world, this can be very complicated to achieve, and so leaving a lot of children unprotected. So a goal in the end here would be a deeper understanding of infant immunity and the factors that affect it in order to try to understand, aid in improving vaccines, vaccination schedules, increasing adjuvants, et cetera, and assessing risks to infectious disease, which is a major goal of many individuals in the global health field. And so, as my pediatrician friends tell me, infants are not little adults, especially in terms of the immune system. And so there are a number of different things that are quite significantly different in the infant immune system compared to the adult immune system. And so we'll start here with, in the upper right is sort of the humoral immunity. So B cells are an important part of humoral and durable immunity, production of antibodies, and specifically in infants, there are a number of defects such as a lot of immature transitional cells, smaller production of high affinity antibodies in germinal centers, and that is partly precipitated by less help by T cells for B cells. And then with regard to the T cell side, there are a lot of immature thymic cells, immature T cells that are present in the infant circulation, which results in is associated with a lower ratio of sort of Th1, which are cells that are capable of fighting intracellular and viral infections, and Th2 being very important for bacterial infections, etc. And this ratio being in balance is necessary for, in addition, protection from infection, but also reduction of autoimmunity and allergy later. The innate immune system is also affected, and so particularly with its ability to stimulate these Th1 responses. And so on the bottom end of this spectrum here are sort of external environmental factors. All these things can affect in the development of infant immunity. And so there's, the premature birth or the season in which you're born, whether it's the wet season or the dry season, in utero stresses, the early postnatal environment, maternal antibodies that mom is transmitting through breast milk or during the first 6 months after birth through transplacental IgG can also have a negative impact on the ability of an infant to mount a strong vaccine response. And the one that I'll discuss is one that Dr. Manary introduced, is nutrition. It's an impact on the immune system. And so malnutrition in and of itself increases a child's risk of dying from infection by about 4 to 10-fold. And work from us and others have shown that malnutrition can be associated with an increase in pro-inflammatory biomarkers and can be associated with lower titers to important vaccines like the oral polio virus, and also, again, this increase in pro-inflammatory cytokines. Yet the conundrum with this is that in the scope of things, most kids are moderately malnourished. It's sort of a Poisson distribution where there's a lot of moderate malnutrition and, of course, some extreme malnutrition. And in moderate malnutrition, which affects the most children, the humoral components, the total amount of antibody present, the accessory factors that help antibodies mediate their function, which is complement, immune cell numbers, these are all relatively resilient to moderate malnutrition. So there's the hypothesis here is that these cells may be less poised at baseline or able to act when are faced with a insult in the context of malnutrition, and that's what we're going to be looking at today. And so the aim here is really to determine whether a transcriptional profile in blood can reveal a distinct baseline gene expression signature that's associated with malnutrition. And so the approach, what we have from a large field trial, is whole blood from 1-year-old infants, and we have separated them into groups, infants exhibiting a control growth trajectory versus an impaired growth trajectory, and are going to be looking at whole genome transcriptomic analysis. So I'm an immunologist, and so I really like doing in vitro functional experiments too, and we have some cellular samples from this patient, but they are very limited. You get one shot at that's a very small volume of PBMCs. And so our thought process through this is that the gene expression profile in blood can help inform some functional assays and that we can be doing with the paired PBMC samples. And the long-term rationale is that in identifying some malnutrition-related impacts on gene expression can yield new biomarkers, critical signaling pathways, and new targeted approaches for supplementation specifically in malnourished kids. And some of this would dovetail very well with what Dr. Manary introduced as macro and micronutrient supplementation, et cetera, could be something that we could look at in terms of the modulation of these critical pathways. And so the project that we're working with is the performance of oral and rotavirus and oral polio vaccines in the developing countries, so the PROVIDE study. And the bulk of this took place as a birth cohort of about 700 children in the capital of Bangladesh called Dhaka. So these children were followed from birth to 2 years old with clinic visits, the diarrhea etiology and severity monitored routinely, immunogenicity of these oral vaccines as well as other systemic parenterally administered vaccines, peripheral biomarkers. I'm using an outdated term. It should be environmental enteric disorder, as Mark Manary alluded to, but environmental enteropathy was the former name of that. Nutritional status is indicated by anthropometry primarily, how well the kids gain weight or gain stature, and maternal and socioeconomic factors as well. And so, just as a brief overview of what I mean by growth here here's an example of on the left is the weight for age z-scores that the WHO has available for the world, essentially. It's a one-size-fits-all, although there are country-specific graphs that are available. And so basically the way this works is the age on the x-axis, the weight in kilograms on the y-axis, and then these dotted lines are standard deviations above the median growth that's been averaged over children across the world to these specific ages. And the traditional definition is this red line here, where the WAZ dropping below a score of -2 standard deviations below the median is the typical definition of malnutrition. And in the PROVIDE cohort on the right, we'll see WAZ as well as height for age z-score and weight for height score. And so in orange here, malnourished kids, the WAZ of less than -2 is about 25% at about 1 year of life. Kids who are stunted, so HAZ score of less than -2 is about 30% at 1 year of age, and children exhibiting wasting disease are about 8% of our cohort with a weight for height z-score of less than -2. And so we wanted to then investigate whether malnutrition it can be correlated with any of the biomarkers, et cetera, vaccine responses that we've looked at. So this is a recent publication where a figure from a recent publication where we measured here on the vertically grossly put into vaccine responses. And on the bottom here, factors such as biomarkers, anthropometry, and background factors. And so these include serum cytokines, ferritin, an indication of iron uptake, calprotectin was mentioned, myeloperoxidase here, Reg1b, gut health, some micronutrients such as vitamin D, zinc, C-reactive protein, soluble CD14, which is a marker of LPS or bacterial infection, and then also some of these other factors such as maternal height and weight, the height and weight profiles at an early time point, and then correlation with titers and responses to common vaccines such as measles, pertussis, diphtheria, poliovirus, rotavirus, and then also here was another way of measuring growth, which is the change in height or weight over the first year of life. And so what we found is that when we looked at the weight and height, we found through this smoothly clipped absolute deviation score, which is a statistical method that is designed to maximize the impact score of significant biomarkers and minimize those that are only tangentially related. And so what this the blue color refers to a positive association of two factors, and the red indicates a negative association. So the way this works here with height is you see some blue popping up with pertussis, diphtheria, tetanus, oral poliovirus 2 titers, etc. And then another, just trying to use my mouse here to advance the slides. Oh, here we go. And so on the in the other direction, if you look at the absolute change in height or weight over the first year of life and identify factors here over here on the left in red, so these are all negatively associated. So what this means here is that if there's a low weight or height at vaccination, this is associated with reduced polio, diphtheria, DTP vaccine responses. And if, on the other hand, if there's a low rate of change in height or weight during the first year of life, there is an association with increased pro-inflammatory cytokines like interleukin-1 beta, and also the biomarker C-reactive protein, soluble CD14, and then ferritin, which is maybe a metabolic indicator, which may indicate some sort of generic infection going on. And so with having identified an association of malnutrition, inflammation, and immunity, we then proceeded to the whole blood samples to get more insight into the systemic immune system baseline. And so for blood, of course, it's a familiar and routine procedure, can be termed a liquid biopsy, where there's longitudinal sampling possible. It hosts a wealth of information, including immune and inflammation markers, and as well cells and also other soluble mediators. The challenge with gene expression in whole blood is that what you really are interested in, or at least I am as an immunologist, are really the white blood cells, so these leukocytes here. So that's where most of the functional information will be available, but leukocytes are about 1,000 times less abundant than erythrocytes and 100 times less abundant as platelets. And so again, similar to what Mark was describing in stool with bacterial ribosomal RNA being the predominant feature, even in blood, where there is really no other foreign contaminant, it's really fighting with between erythrocytes and platelets. And so the isolation, the way you could get around this is the isolation of the PBMCs, but that's not always routine in field settings and can be often difficult in small pediatric samples. And some of the challenges of whole blood without doing isolation of PBMC is that erythrocytes in particular, very high levels of hemoglobin, alpha beta, mRNA, which are highly stable due to modifications in the untranslated regions. And in fact, some recent papers using RNA-seq have been shown to where 50 to 80% of the reads are derived from globin RNA, so you lose power. So this was a new procedure for us, and so our approach was to take in our pilot study 30 control subjects, 30 malnourished subjects, whole blood at 1 year of life. They were gender balanced. We use the HTA 2.0 arrays, and then I also have a outline of how we put our batches together, since we could do about 10 microarrays per day. We didn't want to have batch-to-batch variation, and you could review that at your leisure. But as Mark indicated, the HTA 2.0 arrays give you whole genome coverage, alternative splicing, and at the University of Vermont, we've identified that HTA 2.0 has similar sensitivity in assessing fold changes in gene expression compared to RNA-seq. It's much more cost-effective, to be quite honest. And in fact, our pilot study with only 60 subjects was underpowered for single nucleotide polymorphism analysis. And so all of this led us to HTA 2.0. And so the first step was we did our isolation. In the field, what was done is a procedure that was designed to maximize RNA stability. So actually quite a lot of trizol was used in the field preservation, where as it should be a 3-to-1 ratio, this was about a 9-to-1 ratio. And so this was a little bit different. So first, the whole blood was resuspended in trizol and then frozen at -80. And then on the left here, we did it with a column prep, typical QIAGEN column prep, and then on the right was standard ethanol precipitation. And so the way this was done was thaw the blood sample in the water bath, adding 1 ml of RNase DNase-free water to try to decrease the contaminants that are present in whole blood, removing a couple of aliquots, and then comparing the yields by nanodrop. And what we found here was that the yield was higher after ethanol precipitation and actually resulted in a slightly cleaner profile. So this was, ethanol precipitation was routine and able to provide enough RNA for us to work with. So next, this is a 2-year study. And so the samples could have been stored for quite a long period of time. And so we wanted to test whether the stability of the samples was affected by the storage time. And so here we looked at the nanogram per microliter, or yield, and found no difference between samples that were stored over a year to compared with less than 6 months. In the middle is the RNA integrity number analysis, which, if you remember old school northern blots, basically is a in silico way of looking at the 28S, 18S, and 5S ribosomal RNA as a marker of RNA integrity. And a score of 6 is considered acceptable, and we found no difference in the quality depending on storage time. So what we started then getting into our sample bank, and we found that this in control versus malnourished kids, we had no significant difference in the amount of RNA we achieved, and it was over this line, this dotted line on the left, which was the amount you have to put in for a WT plus target prep kit. And as we gained familiarity with the procedure, we also identified that they're a very high RNA quality, as well as measured by the RIN score. However, and so with the WT plus target prep, which is what we used, was advertised to use 50 nanograms of RNA, and then through reverse transcription, and then in vitro transcription, and then labeling and hybridization to the target prep was supposed to be touted to be able to use whole blood. And so however, in our hands, we found that with our RNA isolation process, that the first step here, this first purple arrow, which is the first C complementary RNA purification quantitation step, you require about 834 nanograms per microliter to proceed, and we weren't able to achieve that. And so we were kind of stuck there. And so what we tried then is a competitor's kit. And so we were able to get some target prep onto the HTA 2.0 chips, but it was quite dim, as you see here, from the PM mean score. So when we used the NuGen, we only had a PM mean score of about 131, compared with using that kit on HTA 2.0 arrays. In a cell line, you get over 300, and if you use the Affymetrix WT Plus kit on a cell line on an HTA 2.0 chip, you also get a robust signal. So the downside of this was that we found a domination of globin transcripts, hemoglobin, myoglobin, and all these other ones, despite being able to make it through some of the early QC steps using this alternative prep. So we thought that globin reduction might improve this. So you have to start with globin reduction with about a microgram of RNA. And so to tell you right off, we had a hard time achieving that. And so we, in the few samples where we were able to get enough RNA, we did globin reduction with the goal of getting 50 nanograms of the input. And so what we see here is that whole blood, again, this is that this is that former step with the whole blood Nugen kit with a very dim chip. When we did globin reduction, we improved the signal, but it was still pretty dim. And then using the globin reduced RNA with the WT plus target prep did increase the signal more. But so we did able to get to a chip hybridization with the WT plus target prep. However, the chips were still a little bit dim, and we did find that the globin signal was gone. But as I mentioned, most of the samples had pretty limiting factors. We even tried as low as 20 nanograms of input with the WT plus, but with no success. So then, miraculously for us, Effie came out with the WT Pico target prep. And I won't walk you through all of this, but basically I'll highlight the fact that there's a much reduced input that is primarily due to this amplification step with Taq polymerase. And so we tried this on some of our samples, and we found that we needed to modify just a little bit at this cRNA amplification step, moving from 9 cycles as recommended up to about 12 cycles. But then we started getting good results, and those are presented here, where across our 6 different batches, we had plenty of input RNA, because as I'll point out here, the input is so low that you need for the Pico kit, that this was more than ample. And so again, our IVT, the in vitro transcription step, where you need to hit 834, we had no problem in any of our batches hitting that. And then at the single-stranded cDNA step, that was even more normalized. And so we had good reproducible production of target prep. And so these data really, and so then we finally had all of our 30 controls and our 30 malnourished samples done in 6 batches. They're all able to be hybridized to the HT8 2.0. And so presented here are the relative log probe intensities for all of the chips. And what we found is that there are a couple of outliers, which left us with 57 out of 60 samples are exhibiting within bounds relative log probe intensity, so that they can be compared. And so these are data that are really just a couple of weeks old, so we're still thick into the bioinformatic analysis. And so I'm not able to present that today. But what I can say is we didn't see any batch or group effects, and we were able to get almost our whole cohort here. And so some of the conclusions and lessons that we learned is that we were able to use the WT Pico kit to isolate the 60 whole genome transcriptomes from whole blood RNA that were field preserved, and that globin reduction was indeed not necessary with WT Pico. Our preliminary bioinformatics tells us that globins are nowhere near dominating as they used to be. So they're actually, so the power of these chips is actually expected to be quite good. And so the lessons are really to, optimize the whole blood sample and storage collect before you even begin. And then the quantitation method is important. When we were getting to these limiting samples, when we were working with the WT Plus kit, we found a big discrepancy between nanodrop quantitation and the fluorometric quantitation. And so the fluorometric quantitation was much more reflective of of the inputs that were necessary. And so the fluorometric quantitation is now standard at UVM. And so our next steps are really to try to undergo a gene set expression analysis to determine functional immune modules, genes that are coordinately regulated within immune cell subsets, and then that will help to inform some more downstream experiments. I just want to thank a lot of people from the University of Vermont Vaccine Testing Center, our Advanced Genome Technology Center for all their hard work in getting this off the ground, our Bioinformatics Shared Resource, Bill Petrie and Caitlin Naylor of the University of Virginia, who were involved, were instrumental for the PROVIDE study, and also the study team in Dhaka who helped perform 15 clinic visits over 2 years on 700 kids, which is quite remarkable. And lastly, I'd like to thank the families for their participation.
Thank you, both Dr. Diehl and Dr. Manary. Now, we're going to open up with question and answers, and Dr. Diehl, the first question is for you. Let's see, with some great questions coming in. Okay, Dr. Diehl, why did you choose microarray instead of RNA sequencing?
We chose microarray because we felt that we didn't really need the sequence information. Our budget was limited, and we used HTA 2.0 because what we were most interested in is information on relative gene expression and potentially alternative splicing. And with just a pilot study of 60 kids, we didn't feel, we did not, our statistics indicated we didn't have the power for single nucleotide polymorphism analysis, which is really a huge asset for RNA-seq. And so that's why.
Thank you, Dr. Diehl. Okay, Dr. Manary, this one's for you. What human cell types are present in feces that you expect to be measuring mRNA expression from? Is this locked off epithelial tissue? Could a population of cells simply be increasing in the RNA sample and not expression of certain genes in one cell?
Right. Excellent question. So, because we really wondered ourselves just what kind of cells we would be sampling and seeing in there. There definitely are epithelial cells from the intestine, and there are definitely a host of different immunological active cells, T cells, B cells, dendritic cells, and macrophages. Are they present in the same proportions as they are in the tissue? Probably not. They probably have more of a propensity for certain cells to shed than others. And we're not particularly, if we could get biopsy samples from, 100 or 200 children, that would really be informative in a different way of this particular condition. But we can't. If you're imagining these are free-living kids who aren't symptomatic and particularly distressed, we can't do endoscopy or anything on them. And just to mention also, the quality of our RNA that we're looking at here is a lot less than what Sean's talking about. So we're looking at RIN numbers of 3 to 5 or so, and he's got a more concentrated sample in his. But yes, so definitely we can find something that looks like a biomarker, find something that looks like pathophysiology from the fecal perspective. Is that the whole story? Probably not. Is that enough of the story to get us on the road to helping these kids who are so stunted? We're hoping so.
I'm sorry, we don't have time for any more questions. I would like to once again thank Dr. Mark Manary and Dr. Sean Diehl for their presentations. I would also like to thank the audience for joining us today and for their interesting questions. We would like to thank our sponsor, Affymetrix, for underwriting today's educational webcast. This webcast can be viewed on demand through April 4th, 2017. LabRoots will alert you via email when it's ready for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Thanks for joining us. Until next time, goodbye.